We also made isogenic mutants that lacked either one or the other of the two proteins that I just described, and so I'm going to describe the characteristics, the phenotypes of those mutants. So we made, so the wild-type strain makes both proteins. We made a knockout of the TBPA gene, and so that mutant doesn't make A, but it makes B. We made a knockout mutation in the B gene, which sits upstream of the A gene. And that mutant, which was a polar insertion, eliminated the expression of both proteins, and that showed us that the two genes are co-transcribed on a single operon. So then we made an in-frame deletion in the TBPB gene, and so that strain makes A but doesn't make B. Okay? So we tested the phenotypes of those mutants, and these first two phenotypes are easiest to think about in terms of the protein that's missing. Okay? So in the A mutant, it's making B, but it doesn't make A, right? And what you can see is the amount of iron uptake that happens in an iron uptake assay is nil. They can't take up any iron at all if they don't make A. And they can't grow on transferrin as a sole iron source if they don't make A. So A is absolutely essential for this process. However, in a mutant that doesn't make B, there's still some level of iron uptake. It's not as efficient, but they can still do it. And they can grow somewhat on iron as a sole iron source. And so that told us that TBPB wasn't absolutely crucial for the process, but it made the process more efficient. And then the other interesting phenotype, which is shown in the next set of data, is easiest to think about in terms of the protein that it remains in the mutant. So in the A- mutant, that strain is still making B, and what we found was the B-only strain now was specifically binding to the ferritin form of human transferrin. It didn't see the APO form at all. So the B protein recognizes that protein, which is useful for iron uptake, right? Because the APO form that doesn't have iron on it, it's not useful. But the A protein that's made in the B- mutant can see both. So the specificity is imposed by the B protein. Does that make sense? Okay. So that was all combined into one of our earliest models of how this system works. So for those of you who are not aficionados of microbes yet, this is a typical membrane arrangement for a gram-negative organism. They have two membranes, the outer membrane and a cytoplasmic membrane. And transport through the outer membrane of large things like iron chelates is generally accomplished through a type of protein known as a TONV-dependent transporter. And they're TONV-dependent because they need this protein known as TONV to energize them. And so that's what's shown in this figure. TONV actually senses the proton motive force, which is a type of energy across the cytoplasmic membrane. And then that energy is handed off to a TONV-dependent transporter, and TBPA is a member of this class of proteins, this TONV-dependent transporter class. And so what I just showed you in the last slide is the phenotype. If we knock out this protein here, the integral outer membrane protein, we get no iron internalization. That makes sense because that protein makes a hole through the outer membrane. And if that hole isn't there, then they're getting no iron, right? The B protein, on the other hand, which is lipid modified, it's stuck out here on the surface, isn't absolutely crucial, but it makes the process more efficient. And until we got the crystal structures, exactly why that was not completely clear. It also, as I showed you, is specific for the form of transferrin that has iron bound to it. So that may enhance, explains part of the reason why it makes the process of iron acquisition more efficient. So speaking of crystal structures, a colleague of mine at NIH just recently, in 2002, published the crystal structures of these two transferrin binding proteins from Neisseria meningitidis, the organism that I told you is very similar to Neisseria gonorrhea. And this is what the TBPA protein looks like. This is a homology model for the gonococcal protein. And in fact, it's shown here in complex with the human protein transferrin. It, like all the other TONV-dependent transporters, is composed of 22 transmembrane beta strands, and those 22 transmembrane beta strands come together to make a beta barrel. And that's what's shown in green. And then inside of the beta barrel, you can see a yellow region, which is actually a plug domain, which folds up inside the barrel and plugs the pore, so nothing can get through there until a signal cascade happens, essentially when the right ligand is bound, then the plug will move out of the way. But the other thing that has to happen is the TON-B protein has to come up in there and pull on the plug. So that's how transport is accomplished through these TON-B dependent transporters. The other thing you can see from the co-crystal structure is that only the C lobe of transferrin binds to the TBPA protein. The TBPA protein is the largest transporter of this class to be crystallized, and most of the extra amino acids are accommodated in these very long extracellular loops, which have a great deal of interaction with this large protein ligand. What's shown here in blue is a little helix finger, and as you can see, transferrin has two lobes, and each lobe is capable of binding an atom of iron, and they're sort of like Pac-Man that are situated with their clefts outwards. And so what it looks like is this helix finger actually reaches into the cleft here and pops the iron out of the cleft. And so we're right now testing that hypothesis by changing these residues and deleting those residues to see what the effect is on the transporter. We predicted the two-dimensional topology of the transferrin binding protein and published this in 2000. And this was based primarily on computer prediction programs and also diversity. If you look at sequence diversity, most of the residues that vary were out here in those extracellular loops. But it turned out now we know the two-dimensional structure from the three-dimensional structure, and actually the two structures are remarkably similar. Likewise, we predicted that there were 22 transmembrane beta strands, 11 relatively long surface exposed loops, and then this plug domain here, which is composed of the amino terminal 160 amino acids. And there were only 3 regions that were slightly modified compared to the prediction. There was one periplasmic loop that was a little bit longer, one extracellular loop that was a little bit shorter, and then another one that had a little more beta strand. But otherwise, they were remarkably similar. Over the years, using that two-dimensional topology model that I just showed you, we created a whole series of different mutants. And I'm only going to point out, and now they've been overlaid on top of the known crystal structure. And I'm only going to point out a few of them because only a few of them had functional defects. And one was, well, a couple of them shown here. We deleted loop 4 and loop 5, and both of those completely prevented binding and uptake functions. So that pointed to this region as being key in the interaction and transport process. We also made an insertion right here where this blue sphere is within loop 3, and that also abrogated function entirely. And it's interesting because that's just a few amino acids down from this helix finger. And we now think that this 9 amino acid epitope insertion actually moves this helix finger out of the right region, and that explains why this mutant was completely defective. We also insertionally mutagenized the plug domain, and likewise had negative impacts on function. We've also done some site-specific mutagenesis of this motif, this EI-EYE motif. And the reason we chose that is because those residues are capable of coordinating iron, and we've hypothesized that the plug domain actually binds transiently to iron during the transport process. And we've got in vitro evidence to suggest that there is a weak chelation of iron by these residues directly. And so this EI-EYE motif is shown here, and that's actually inside of a putative iron pathway through the barrel. And so we think now that these residues transiently coordinate the iron as it moves down through the barrel. And hopefully on the next slide I'll show you a little movie of how this might happen. Here we go. So what happens is the plug domain, as you can see, gets unraveled, pulled out of the barrel. And when that happens, that actually opens up this iron pore through this barrel. And then, as I mentioned, this EI-EYE motif here transiently coordinates the iron as it permeates down through the barrel. And then what happens next is there's a binding protein that snags the iron out as it comes out of the inner orifice of the barrel, and then takes it through the periplasm and brings it on into the cell. colleague Susan Buchanan and her postdoc Nick Moynihan likewise crystallized the TBPB protein from the meningococcus, and its structure is shown here in purple. It also binds exclusively to the C lobe of human transferrin, and it seems to only bind with one lobe, at least in their crystal structures. What's shown on the right-hand side of the slide is, again, we've done a whole series of, created a whole series of mutants, which abrogate the function of TBPB, binding function, and its helper function for whatever it's doing with A. And they largely are localized to this end lobe of the TBPB protein. The Buchanan group also was able to get a tripartite complex. They couldn't crystallize the three proteins together, but they used electron microscopy techniques, electron microscopy techniques, to look at big crystals, or big tripartite structures using these three proteins. So here's the TBPA protein, which is the integral outer membrane protein. Here's the TBPB protein, and here's the transferrin protein. And here's the C lobe of transferrin to which both of these proteins individually bind. And what became clear is that when the three proteins are in complex, there's an iron chamber here that's formed. And so our working hypothesis now is that TBPA, probably through that helix finger motif, pops the iron out of that cleft of transferrin, but TBPB makes the process more efficient because it holds the iron down and prevents it from floating back out by virtue of forming this iron chamber. Does that make sense? And that's the way that iron can be transported from outside the cell down through this pathway that I showed you through the pore, and then ultimately come out the other side and bind to a higher affinity ferric binding protein. So overall, this is what we think is going on with these two proteins. TBPB, by virtue of its ability to bind the ferrated form of transferrin, which is the only one that's useful for iron uptake, it interacts with the ferrated form and then brings the iron and more specifically the C lobe of transferrin to TBPA for stripping. It hangs on there long enough to keep the iron from floating out, and then once the iron's removed, then TBPB actually helps to bump off the transferrin that's now been stripped of its iron, because I told you that the TBPB protein only recognizes the ferrated form. So this specificity helps both in the association and in the dissociation of the ligand from the transporter. Okay, and this is just sort of the status of the field of what we think is going on with TONB and how that charges these transporters. This is the so-called pulling model, where the TONB protein is largely periplasmic, and it reaches up and recognizes a TONB-dependent transporter that's got ligand bound. Actually, the plug is situated in a different place depending on whether the ligand is bound or not. And the TONB protein then reaches up here and grabs what's known as the TONB box. And when that interaction happens, then something mysterious, we still don't understand this very well, but something mysterious then causes a pulling, dragging downwards reaction, and that then unravels the plug, which then accomplishes transport from out here on the outer cell surface into the periplasmic face of the transporter. So this is a transition away from structure and to the vaccine implications. I started with, we're looking for vaccine antigens, vaccine candidates. So what about the transferrin receptor? Is that a good vaccine candidate? Well, the good news is that it's not subject to any of these high-frequency phase or antigenic variation mechanisms that I described to you. Unlike the lactoferrin iron acquisition system, which is not only phase variable, but only half of gonococci have the genes to allow them to use lactoferrin as an iron source. So that's not useful. The hemoglobin receptor system is also phase variable. It's turned on and off, and most of the time in vivo it's off. So it's not a good target. The transferrin iron acquisition system, on the other hand, is always on, always expressed. We know that we can generate antibodies both against the whole protein and against specific snippets of these proteins in animals. sessions, we've looked at mucosal secretions from both men and women who are naturally infected with the gonococcus, and there's little to no anti-TBP antibody. And that's consistent with the fact that there's no protective immune response that's elicited as a consequence of infection. If there was a lot of TBPA or TBPB antibody in infected people, we have to conclude that those antibodies are doing zero good, right? Because these people are not protected from a subsequent infection. And so the question is, can we generate a protective immune response by vaccination? And so that's one approach that we're taking. We've been using the B subunit of cholera toxin as a way to deliver these antigens. It's a very good adjuvant, which gooses the host immune response, and in particular, it's good at goosing a mucosal immune response. And that's really that's the first line of defense. The organism lands on generally the genital urinary tract or genital urinary tract on the mucosal surface, and if we can get a good mucosal immune response there, we might be able to stop the infection right there. So we've generated antigens, both chimeras that have part of the cholera toxin and part of the TBPs on them, and we've also used whole proteins, the whole of TBPAs and Bs, chemically conjugated to CTP. And in both cases, we were able to elicit mucosal and serum antibodies against those proteins. The B response is always way higher than the A response, but the B protein is much more heterogeneous from strain to strain. There's a lot more sequence diversity among the TBP proteins. So A is well conserved, but it's not very antigenic, and B is pretty diverse, but it's very highly antigenic. So we think our best approach actually is to combine the two, because we get the best of both worlds. We get high antigenicity, but we get cross-reactivity between strains. And the mucosal antibodies were actually growth inhibitory. So if we added mucosal secretions from immunized mice, that blocked the ability of the gonococcus to get at transferrin in an in vitro growth assay. So what are we doing now? We're testing the full-length proteins in mixtures in particular. And then I have a colleague at Andrews at Uniform Services who's developed a mouse model of genital tract infection, a female mouse model. And so we've been immunizing mice intranasally with these conjugated proteins, and then challenging the mice intravaginally with the homologous strain, and asking whether immunized mice are more protected than those mice that have not been immunized or have been mock immunized. And the initial experiments are promising, in particular when we use the two antigens together. When we've immunized with both, we get accelerated clearance in mice that have been immunized with both proteins. So it's still an ongoing story, but it looks promising. So what I've primarily talked to you about up to this point is this system, this transferrin binding protein A, and this system which binds human transferrin. I've also mentioned to you that they can use lactoferrin. Only 50% of gonococci have this system. And they also can use hemoglobin as an iron source. And the mechanisms of how this works, neither system have been very well worked out, and none of these proteins have been crystallized. We also know that the gonococcus can use intravactin and other siderophores that are produced by other bacteria, and we know that this FET system is responsible for internalization of those heterologous siderophores. But if you scan the gonococcal genome, it becomes apparent that there are other conjugate-dependent transporters based on the sequence similarity. They all share, again, 22 transmembrane base strands, a lot of sequence homology, and the plug domains are very well conserved. So you can fish these out of genome sequences. And there are 4 that are completely uncharacterized, well, except by us. And so we call these orphan transporters. We don't know what they do. And we call them TDFF, TDFG, TDFH, and TDFJ. And the most closely related transporter to TDFF is a siderophore transporter. And the rest, and likewise for G, the H transporters, H and J, look more like heme transporters. So their possible functions is, again, they might be hijacking xenosiderophores or siderophores from other bacteria, like the FET system does, or they might be internalizing heme which is what their homologs do. Or they might be important in intracellular infection. And so the next slide I'm going to show you is some data we have and published some time ago for one of these transporters in regards to intracellular replication or survival. So this is our model system. So we not only look at iron acquisition outside of cells, but we're interested in how the gonococci gets into cells, epithelial cells, and also survives within epithelial cells. And this is our epithelial cell model. It's a cervical epithelial cell and a moralized cell line. And what I've shown you here are fluorescently labeled gonococci associated with one of these ME180 cells on the left. On the right is a graph that shows the proportion of wild-type intracellular bacteria comparing the mutant to the wild type. And so the way we do these assays, it's a gentamicin protection assay. We infect this monolayer with the gonococci for 4 hours, and then we treat with gentamicin to kill off all the extracellular bacteria. And that's time 0. That's the light purple or the lavender bar. So at time 0, this is how many intracellular bacteria we have. And this is a ratio of wild type. So for wild type, we get 100%. And then we do the same thing after 24 hours. We do another gentamicin treatment to kill off anything that might have popped out and ask how many survive after 24 hours. And that's what's shown in the dark purple bar. And so what you can see here is that if we add an iron chelator, the survival of the gonococcus goes way, way down. So they need iron in order to survive inside these epithelial cells. You can also see in the next column that a TON B mutant, which I've already described to you, this is the energization system for TON B-dependent transporters, is likewise deficient in both the time 0 time point, meaning invasion, but also in intracellular survival. So that suggests that TON B-dependent transporters are important both for invasion and for intracellular survival. And then finally, this last column shows that one of these uncharacterized TON B-dependent transporters, TDFF, is in fact important for this intracellular survival phenotype. So we don't know what this transporter is allowing the gonococcus to do inside of epithelial cells. It might be hijacking some low molecular weight iron chelator that the host provides, but that's where we're headed with this TON B-dependent transporter. And then more recently, we've identified a novel pathway for invasion into epithelial cells, which is iron-induced, and that's what's shown in this figure. So if we don't add iron to the invasion process, the time 0 that I showed you in the last one, we take gonococci, infect them onto these ME180 cells for 4 hours, and then do the gentamicin treatment. If during that 4 hours we don't add iron, we get hardly any invading bacteria. But if we add iron, we get tens of thousands, 10,000, 1,000 to 10,000 fold increase in the number of internalized bacteria. So there's an iron-induced invasion process that this defines. And it turns out one of the TON B-dependent transporters, TDFJ, is really important for this process because when we knock it out, the iron-induced invasion goes down dramatically. There's another protein called NSPA that if we knock that out simultaneously, this amount of invasion basically goes down to baseline. So we've identified 2 contributors to this iron-induced invasion, and one of them is in this class of TON B-dependent transporters. Okay, so with that, I'm going to conclude and tell you where we're headed in the future. I have shown you the structure of the gonococcal transferrin glycoprotein A, and it's comprised of 22 transmembrane beta strands and 161 amino acid plug or hatch domain that is crucial for transport of the iron into the cell. We propose in our testing right now whether this helix finger in week 3 is important for iron extraction from the C lobe of transferrin. Both TBPA and B bind exclusively to the C lobe of transferrin, and the B protein doesn't interact with TBPA in this tripartite complex, but it does form this iron chamber that very highly conserved. So the TPA proteins from the gonococcus are very highly similar to those of the meningococcus. And so it's conceivable that a vaccine that's comprised of these proteins might not just protect against gonococcal infections, but also meningococcal infections. That would be sort of the best case scenario. And I've also introduced you to a couple of the other TAMI-dependent transporters, and two of them seem to be important in aspects of host cell infection. Next to last slide is a list of acknowledgments. The folks on the left-hand side are postdocs and students and others in my laboratory who contributed directly to the research that I presented to you today. The folks on the right-hand side are folks who have been in my lab and currently are in my lab, but they can contribute directly to this project, with these projects. And then Al Crumblis in the chemistry department at Duke helped to do the biophysical examination of this EIEYE motif. And Susan Buchanan and Nick Wainage at NIH did the crystal structures. And as I mentioned, Anne Jost, my colleague at Uniform Services, has the mouse model. We collaborate a lot with them. And then obviously funding from NIAID supported this work. And then lastly, I wanted to